People from India have taken permission that they will not be around today because of some reasons. And so we are now looking at the time of day for sampling. What time of the best time of day for sample collection in microbiology research? The best time of the day. You see, it's not just any time that you take sample. You go and just take sample anytime you feel like, just go and collect sample. By the time you go and be collecting the sample, when you come to your defense and they ask you and you miss it, everybody will shake your head there. They'll be looking at you like this. They'll be looking at you, they'll be looking at you. They'll be repeating you because they can ask you to go back. They can ask you to go back. Normally, you are supposed to do a number of seminars. You now do presentation. You now miss, you now mention, you now see that there's problem in your sample collection. They ask you to go back and take sample. So to avoid that, that's what we're talking about sample collection now. So that everybody can know and get set. If you miss sample collection, every other thing you have missed it. So now we've talked about different sample collections from the soil, from water, from wastewater, from every other thing. Now we want to look at the best time of day for sample collection in microbiology research. It is not any time you like that you go and take sample. So in microbiology research, the timing of sample collection can significantly influence the quality and reliability of the results. Microbial activity, physiological changes, and environmental conditions often follow diurnal rhythms. That means day and night. So in the day, there's a way the organisms will react. In the night, there's a way the organisms will react. That's what we call diurnal. That means in the day and in the night. So they follow like that, making it essential to carefully plan sample collection times. So this webinar explores the importance of timing in microbiology research and provides insights into the best practices for optimal sample collection. Now, why timing matters in microbiology sample collection? The first reason why timing matters in microbiological sample collection is because of circadian rhythms of microorganisms. Microorganisms have a circadian rhythm. So many microorganisms, including bacteria and fungi, exhibit circadian rhythms. That is, biological processes that follow a 24-hour cycle. For example, certain pathogens may be more active or abundant during specific times of the day. Not just pathogens, certain bacteria in water, certain microbes in soil, certain microbes in any kind of other environment may be active during a particular time of day than the other time, and that can impact their detectability. Number two reason why timing matters in microbiology sample collection is because of host factors. These host factors in clinical microbiology. The physiology of the host, human, animal, or plant, often influences microbial populations. For instance, hormone levels, immune responses, and metabolic activity fluctuate throughout the day, potentially altering microbial composition. Number three reason why timing matters in microbiology research is because of environmental influences. Environmental samples such as soil, water, or air may also vary by time of day due to changes in temperature, humidity, and light exposure. These factors can affect microbial growth and behavior. Another reason why this is important is because of consistency and reproducibility. Collecting samples at consistent times, maybe in the morning, early in the morning at 8 o'clock, 8 a.m. So when you say you take a sample at 8 a.m. in the morning, somebody else wants to reproduce that experiment, that research. The person will also go by what time? By 8 a.m. Let's say your own research was by 8 a.m. Somebody else may say, okay, this person has taken sample by 8 a.m. Let me take by 9 a.m. and be able to compare what 8 a.m. got and what 9 a.m. got. But if you're just taking sample any time of the day, it makes it will make that work not to be reproducible and not to be consistent. Then we now look at the best times for sample collection in different contexts. Number one, in clinical microbiology, from blood samples, blood cultures for detecting sepsis or bacteremia are best collected during fever spikes when bacteria are most likely present in the bloodstream. So that is when they take it. That's the best time, when the spike is high. For urine samples, first morning urine is ideal for detecting urinary tract infection, as it is more concentrated and likely to contain higher microbial load. So that's for urine sample. Early morning urine will be more concentrated than other urine sample. Who is this that is not muting his this thing now? So I was talking about clinical microbiology, best time of the for sample collection in different contexts. For blood sample, when the bacteria, when the when the infection is higher, when the fever is spiking, that's the best time because there's higher concentration of the causative agent in the system. Then for urine sample, early in the morning, because by that time the person have not gone to urinate throughout the night. So in the morning there's concentration. Then for respiratory samples, early morning sputum samples are also recommended for diagnosis. You can see all of the early morning. You can see. So early morning, during that night, is that about activity is not much. So if you really want to see what is actually going on, early morning you can be able to see that. So for respiratory samples, like collecting sample for tuberculosis and others also, it is that time of the day that you can do that. Now, in environmental microbiology, you can take your sample, water sample. Sample should be collected in the early morning or late evening when water bodies experience minimal human or animal activity. So that is when you take water samples, early morning or late evening. But I said it initially that why should you take sample in late evening? What are you going to now be working the lab from late evening, and then there's danger if you take sample in the late evening? If the kind of water it is, somebody can be attacked by any kind of animal in the night, depending on the area or the location. But early in the morning, it is difficult for you to be affected. So that is that. Then for soil sample, mid-morning is often ideal as soil moisture and microbial activity stabilize after overnight cooling. So this soil sample also early morning is also the best. Then talking about air sample now, these are all environmental sample. Air sample sampling in the early morning can capture microbial population before they are affected by daytime temperature and wind pattern. Early in the morning for air sample, the wind pattern is not disturbing them, so you can easily take your sample and it will be representative. So you can see now that word again. So early in the morning, in most cases, you can get representative sample. Now, in food microbiology, when studying spoilage or contamination, samples should be collected at various stages of the food production or storage process. But early morning collection often provides baseline data. You can see early morning. Even in food, if it is in the afternoon, the sunlight rays and the UV light is going to affect the microbial load. That's why early morning is the best time. So early morning is the best time for microbiology. If you want to really do residual work in microbiology, get to the lab early in the morning and then start your work. And before you know it's afternoon, you are off. The following morning again, early in the morning, you are in the lab again. You do your work, by afternoon or evening, you are off again. You leave the lab, except even some most times if you start your work by 8 a.m., before 12 or before 11, you are off, you are done. And you have to plan the work that you want to do appropriately and make sure everything is in order. Then for research studies, for experiments involving microbial cultures, samples should be collected during the exponential growth phase to ensure active and viable populations. Now, if you are even doing biodegradation studies and you want to grow your bacteria broth that you want to add to the reaction mixture where you have the mineral salt medium, you have your hydrocarbon, and now the organism. The organism must be at the exponential phase. That is when you add the organism to the sample so that It will pick up. Not that you go and add the organism at the lag phase. Then to take from lag phase, it will now go to log phase. You understand? So you add it at the exponential phase so that it will get into the reaction mixture and carry on its work. Now, the following are the factors to consider when planning sample collection. Please, Anolua, mute yourself, please. Mute yourself. So the following are the factors to consider when planning sample collection. Number one is the objective of the study. What is the study for? So the objective of the study will determine how you're going to take the sample. We'll talk about that in water. Yes, normal water sample should not be filled to the brim, but it should be like 75% or 80% of the bottle. But if it is involve you doing BOD, in fact, the sample for BOD should be collected separately. The bottle for BOD collected separately make it separate from the one you want to use for microbiological analysis. So if you are going for sample collection and involve BOD, that bottle should be separated from the one for the microbiology analysis, so that there will not be any story. And you should do the day one. Immediately you come out of the lab, you do the analysis for day one. It doesn't take up to 30 minutes to run the test for the dissolved oxygen in the lab. It doesn't take 30 minutes or so. And then you completed that, you get your result. By day 5, you repeat the same thing, and then you get your result. Then another factor to consider when planning sample collection is sample type. So different type of sample require different type of collection system. Then you also have to consider the storage and transport. How do you want to store the sample? How do you want to transport the sample from the point of collection to the lab? If it's far away, you have to use ice chest. If it's not too far, you may not use ice chest. If you are going to be away in the afternoon, you have to use ice chest. But sometimes I see people coming to the lab with their sample by 3 o'clock, by 2 o'clock, and I wonder, where did you collect the sample? That means you collect the sample 11 o'clock, 12 o'clock. Ultraviolet light or sunlight has already affected your sample before you collected it. So all these are the things that we should look into. So if you have so many sampling points, maybe you should collect 2 at this particular day. Another day, collect another 2, so that you don't need to be going from here and then go far to the next place. It is not compulsory you take all your samples in one day. You can focus on just one sample point today. Tomorrow, another sample point. Next tomorrow, another sample point. But most people don't want to do that. And then what you get is that they start sample collection by 10 a.m. Then by 11 a.m., they're in the sample collection point B, then place B. By 12, then place C. By 1, then place D. You can see that sample they're collecting in the afternoon or around that time is not representative, is not ideal. So another factor to consider when planning sample collection is the environmental condition. So you monitor and document temperature, humidity, and other environmental factors. For this, you have to have your thermometer with you. So as you're in the sampling point, you check the thermometer of the environment. As you're in the sampling point, you check the pH of the sample directly where you are taking the sample. So you go with the handheld pH meter and other humidity meter also. And for other environmental sample, you go with them and you check your sample and you write it down immediately. And then that is going to be easy. And in most cases, you don't go for environmental sample alone. You get somebody that will help you to do some things, carry some things, move something. While you are doing this, you collect it, you give the person, you do this. So it makes it easy for you. Then if you are taking measurement on the field, the person can be writing for you. As you are taking, the person will write. As you are taking, the person will write for you. And that is what makes it easier and ideal. And in fact, the person in that case will be learning how environmental samples are taken. And then that is even much more than any classroom knowledge that the person can get. So the following are the tips for effective sample collection timing. Number 1 is to plan ahead. Number 2 is to use control. In every experiment you do, use control. Includes time-based controls to account for potential variations. Then number 3, document your timing. The time you took your sample, document it, write it down. Then record the exact time of collection for each sample to facilitate accurate analysis and interpretation. If you don't document properly, what you got may be right, but you got it by 12 noon, but you know that you got it by 9 a.m. So it's going to affect your interpretation, your discussion, because some things that are not supposed to be, you'll be saying that they are, because you took your sample by what time? By 12 noon, and you wrote 9 a.m. So these are some of the things. So you also have to train your personnel. If you want to go with somebody, you have to train the person on how to do this, how to write this, how to do this. And usually you go with somebody that is a microbiologist like you, but maybe it's younger maybe it's somebody that is a younger microbiologist, so you go with you. And then in fact, in that case, you don't need to train the person. But in some cases where you are using a non-microbiologist, you have to train the person and say, okay, this is what this means, this is what this means. And that will help you to do what you want to do on the field and take all your samples, and then you get back to the lab, and then you continue your analysis. So these are the ways for sample collection and that I've looked at. So I don't know if you have any question. So this theme of this webinar is sample collection in microbiology. So if you have any question on this, you can ask before we close or we adjourn the webinar for today. If you have any question, you can ask your question before we go now. So I can see that there's no question. So in the absence of any